Hello, my name is Jesse Larson, and I'm an ATA for Bio 201, Anatomy and Physiology, and I'm the ATA for Autumn Irvin, who is my TA. Today we're going to go over the parts of the microscope. The microscope was invented in the 1400s and allowed us to see more into the smaller cells and allowed us to progress science much farther. This right here is called the arm, and it is connected to the base. Whenever you are transporting the microscope, you always want to have one hand firmly on the arm and another hand firmly underneath the base so that you won't drop it or anything. And this is the cord. It needs to wrap around the base so that it doesn't snag on anything and won't rip it out of your hands. Up here we have the ocular lenses. This is called a binocular lens because there are two lenses. And then on the older styles, this is called a monocular lens because there's only one. Right here we have the objectives, which we have a 10x, a 40x, and a 100x. You only want to use a 100x when you are told to by the TA because you'll have to use oil. But to be able to see the specimen on the slide, you need to have light. And so this is called the rheostat, which you turn this wheel and it allows light to come in to the specimen. And on the older versions, you twist this wheel to allow the light to come in. Underneath the stage, we have the iris diaphragm, which allows you to allow more light in or less light in, depending on what you're looking at. Okay, to continue on, to move the stage closer to the objectives, you use the coarse knob, which brings the specimen a lot closer so that you can see the cells. And once you see the cell, then you adjust the fine focus knob so that you can see more of the intricacies of the cell to be able to know what you're looking at. And when you bring it closer, it's going to be magnified 10x through the objective and also another 10x through the ocular lens, and so it's going to give you a total of 100 times magnification, which is very important when you want to see very small parts of the cell. To be able to put a slide in to the microscope, you move this clip and the clip will fasten it into the microscope. And to be able to move where you want to see on the lens, you can move these knobs to go right or left or up or down. And basically that's the main parts of the microscope. Okay, now we're going to go on to part 2 and we're going to learn how to prepare slides. In Bio 201, sometimes we have prepared slides that are already made for us, but sometimes we're required to make a wet mount slide, and so that's what we're going to do a little demonstration on blood. And so since we're using blood, I have gloves on so that we don't get blood on our hands. But for demo purposes, we're just going to use some red food coloring. So I have a slide right here, and then I also have a cover slide that I'm going to put on top of the blood drop. So I pipette some up into the pipette, and just a little drop. Now when you take the cover slide, you're going to want to put it on the corner right here, and once you see it, grab it like that and make a line, you just drop it. And so this slide is prepared to, actually it's not. If you have some excess blood, you can kind of dab it from the corner so that there's not so much blood on there. And then you're prepared to go on to part 3 with looking through the microscope to actually be able to see the cells. Okay, all the information that we've gone over previously would be pointless unless we can actually see what's on the cell. So you turn on the microscope and clip in the slide and kind of move it to a corner so that you can see parts of the cell. And you want to adjust the light because you don't want to white out the cells that you're looking at. So you can adjust the light by using the rheostat to dim it, or you can adjust the iris diaphragm, which would let more or less light in. So kind of experiment with what works best when you're looking through the microscope. And then you'll use the coarse knob to move the stage all the way up to the objective. Keep in mind it has to be on the lowest objective first. And also, this is a 40 magnification because there's 4 by the objective and then 10 by the binocular lens. So that's a total magnification of 40. Then once it's all the way up to the top, then you move down until the cells come into view. Once the cells come into view, then you use the fine focus knob to see more of what's inside the cells. Then, once you're done with that, then you move to a higher objective so that you can see even more of the intricacies of the cell. After you're done, move the stage all the way down using the coarse knob. Turn off the rheostat and wrap the cord around the back of the microscope. Then remember to use the arm and the base to transport it to where it needs to go if you're putting it away. Then there's either 2 ways you can clean the slides. You can either wash it off and put it back at your station, or you can put it into the disposable glass container. Basically just listen to your TA and follow their instructions so that you can do this all properly. And this is basically how you use a lab, or how to use a microscope in a lab.